last time talking about the Wittig reaction, and this was part of a couple of reactions at the very end of chapter 14 that were reactions that didn't necessarily fit with the main theme we were talking about, but which nonetheless you might encounter in future standardized exams, so I wanted you to be prepared to at least recognize the reaction and know how to use it. The Wittig reaction is a Nobel Prize-winning reaction between an aldehyde and a ketone, or a ketone and a reagent called an ylide, or a Wittig reagent. And the process makes a new carbon-carbon double bond between this negatively charged carbon of the ylide and the carbonyl carbon of your aldehyde or ketone. And you also make this triphenylphosphine oxide as a product. This is very stable and drives the reaction forward. Where stereoisomers are possible, you get both, and we won't worry about controlling E versus Z. I won't hold you responsible for the mechanism of this reaction, though I do want you to know how to use it in a synthesis or be able to predict the products or provide the reagents. And so I will talk through the mechanism because sometimes I feel like if I don't see the mechanism, it's harder for me to remember what the reaction does. You can make the Wittig reagent by taking a phosphine, which is what you call a phosphorus bonded to 3 carbon groups, in this case benzene rings. The phosphine can be a nucleophile. It has a lone pair just like an amino group can. You can do the SN2 reaction where phosphine dumps its electrons into the carbon-bromine antibonding orbital and breaks the carbon-bromine bond. And that makes this new reagent, this new product known as a phosphonium salt. A phosphorus bonded to 4 things is positively charged, just like a nitrogen bonded to 4 things is positively charged. NH4 plus would be an ammonium group, so a phosphorus bonded to 4 things is a phosphonium group. It's a salt because it has a counter ion, which we don't need to worry about. Then you take a very strong base. This is an organolithium reagent that's also a really, really strong base. To remove the proton adjacent to the phosphorus. That proton is more acidic than any of the other sp3 hybridized carbon-hydrogen bonds because of its proximity to the positively charged phosphorus. And that generates our Wittig reagent, which is called an ylide. And you don't need to remember that term, but in OChem, that's what we call a molecule that has a negative charge and a positive charge adjacent on adjacent carbons. Now this reagent is nucleophilic on the negatively charged carbon. Your text, I think, shows you an alternative resonance structure. Some people prefer to draw it this way, with the electrons on that negatively charged carbon kicking down to form a new carbon-phosphorus double bond. That might give some of you heartburn because the phosphorus here, if you'll notice, is bonded to 5, making 5 bonds, which for second row elements was a big no-no. Phosphorus is 1 row down on the periodic table and is bigger and so can form more than 4 bonds. We're not going to talk about the orbital picture that makes that possible. We'll just accept it as a given. But I prefer to use the ylide resonance structure because it helps me remember what comes next, where we're going to use the negatively charged carbon as a nucleophile. So this is the Wittig reagent. You have the alkyl group and the phosphorus group. Next step is going to be for the negatively charged carbon, the nucleophile, to attack the carbonyl carbon, to attack the pi star at the carbonyl carbon, thereby breaking the pi bond. And so far, that's just exactly what we've seen before for carbonyl chemistry, the addition step. Let's attack the pi star to get a tetrahedral intermediate. The one difference is going to be that we have a phosphorus nearby, and phosphorus-oxygen bonds are particularly strong. And so instead of making a negatively charged oxygen, we're going to actually have lone pair electrons on the oxygen attack the phosphorus. Again, that's going to seem weird because phosphorus is making 5 bonds, and we're just going to have to accept that as a given. but it's possible because of the strength of phosphorus-oxygen bonds. So here are our 3 phenyl groups on the phosphorus, and then here is the other alkyl group. Here's the 2 new bonds that we made. All right. This is a 4-membered ring intermediate, and your text names it, but we don't need to worry about the name of it. What happens next is driven by the fact that oxygen-phosphorus bonds are so stable. Electrons in this carbon-oxygen single bond will actually rearrange, move over to form a new phosphorus-oxygen pi bond, thereby breaking the carbon-oxygen bond. At the same time, electrons in the carbon-phosphorus bond will kick down and form a new carbon-carbon pi bond. On the phosphorus side, you make triphenylphosphine oxide, which is really stable, and it's the stability of that product that actually drives the reaction forward. And then you get the new carbon-carbon double bond plus any stereoisomers if possible. So this reaction you can think of as sort of a Lego reaction in that you're clicking together the carbon of a carbonyl group with the negatively charged carbon of the ylide, and you're making a new carbon-carbon. You're making 2 new bonds, one carbon-carbon sigma and the other carbon-carbon pi. Okay? Questions about what happens in the Wittig reaction? Okay? You can think of this in some ways as the reverse of ozonolysis. I don't know if you remember in 351, if you learned ozonolysis, it splits up a double bond into 2 different parts, and both of them are carbonyls. This is sort of like the backwards version of that, in that you take one carbonyl and then the other partner is not a carbonyl, but you stitch it together to form a new double bond. So you can sort of think of it that way. Your text will want you to be able to think about this from a retrosynthetic point of view. So if I have this alkene, how would I have synthesized it via a Wittig reaction? If one of the 2 carbons involved in that carbon-carbon pi bond only has one alkyl group on it, you want to choose that as the ylide portion and this as the carbonyl portion. And the reason for that has to do with the 1, 2, 3, 4. It has to do with where the Wittig reagent comes from. Remember, you originally make that from an alkyl halide and a phosphine. So SN2 reactions with primary alkyl halides are fast. And so if you can, you choose the side where you can use a primary alkyl halide to make the Wittig reagent. So be able to use that reaction and predict the products and provide reagents and synthesis, but don't worry about the mechanism. All right, anything else on that? Okay, so we're going to move on to chapter 15, and talk about acids and bases. Much of what we talk about is going to be review from 351, so we're not going to spend a huge amount of time on it. But there is an important principle that I think will be useful to you in future biology courses, which we will talk about in more detail. Just briefly, you know that a pKa tells you how acidic a particular proton is. So, for example, acetic acid has a pKa of 4.8. Don't memorize that. Whereas alpha chloroacetic acid has a pKa of 2.8. So, which is the more acidic compound? The chloro one, right? Okay, good. And the reason for that is, and whenever we rationalize acidity, most frequently we draw the conjugate base and compare the stability of 2 conjugate bases. Both of these have the negative charge stabilized by resonance on both oxygens, so that's not the difference. The key difference is the presence of a electronegative chlorine, which pulls electron density away from the alpha carbon, which in turn pulls electron density away from the carbonyl carbon, which in turn can sort of pull electron density away from the negatively charged oxygens. This is called an inductive effect. It's called electron withdrawal via an inductive effect. Another example of electron withdrawal, but this time via a resonance effect, is comparing the acidity of the molecule on the left, which is called phenol. Don't ever call it phenol. It's just like it's irritating to organic chemists. I suppose if you want to make us angry, you can call it phenol. But I just remember being totally put off in a biochem class I took here 100 years ago when I was a student, and we were using phenol and chloroform to do a DNA extraction, and people don't even do that anymore because there are kits, but it talked about, we kept being told you're going to do a phenol chloroform extraction, and it was like nails on the chalkboard to me. But I didn't say anything. I tried not to be that guy in the class. Actually, it's okay. So the pKa of phenol is like 10. Oh shoot! I should have written down the pKa. The pKa of paranitrophenol is around 7. Don't hold me to that, but it's in that range. Okay, and if you ask why, it's probably not electronegativity of that nitrogen, because that nitrogen is way far away, and the inductive effect drops off as you get further removed from the acidic proton. So let's draw the conjugate base. In both cases, the negative charge on the oxygen is stabilized by resonance, and you should be able to draw resonance structures where you move that negative charge to these positions around the ring. And that's true for the nitrophenol as well, except for the presence of an additional resonance structure that looks like this, where we're basically moving the negative charge from the oxygen out onto one of the nitro group oxygens. And that's how you get from the resonance structure on the left to the resonance structure on the right. So this is electron withdrawal via resonance. And so just be aware of those and be able to compare acidities of things and rationalize your way through them. Anything you want to ask about that? Yeah?
So, if I understand correctly, if you're like a phenol, you have weaker attachment, that becomes an electron donating group. Why is it electron donating for resonance, but it would be withdrawing?
So I think the question is, what if I had a paramethoxyphenol? I don't know what that pKa is, but I know it's greater than 10. And I think what you're noticing is that, so this is less acidic than regular phenol. And so why is that the case? You do have an electronegative oxygen that through an inductive effect can pull electron density out of the ring. At the same time, lone pairs adjacent to pi bonds can participate in resonance. So if we draw the conjugate base of paramethoxyphenol with a negative charge, excuse me, lone pairs on that oxygen can actually donate into the ring, and that puts extra electron density adjacent to the negatively charged oxygen, and that is a destabilizing effect. So yes, there are some functional groups that have offsetting effects. They're electron withdrawing via electronegativity, but donating via resonance. We're going to learn a whole lot more about that later and learn which groups are dominated by their resonance capabilities versus others. But yeah, for right now, we won't worry about that too much. Other questions? Okay, so now it's time for some math. My favorite part of the lecture is where we get to derive equations. Actually, you don't need to know the derivation, and you actually don't even need to know the equation as long as you understand or remember the principle. You encountered the Henderson-Hasselbalch equation in general chemistry, probably not in 351 maybe, but you probably use this to answer questions like this. I want to make a buffer that has a pH of 6.5. How much of this acid and its conjugate base, in what ratio should I mix the acid and the conjugate base to get that particular pH? And it was at that point just math, and you plugged it into your calculator and you found that X grams of this particular acid and Y grams of the conjugate base would make a buffer with just the right pH. That's fine, and that's useful if you're a tech in a biochem lab making buffers for grad students to use. But that's not going to be most of you. So when would you need to use this equation? What else is it good for? And the answer is it's useful for asking about biological molecules, which acidic protons are still going to be present versus gone at physiological pH. So I'm going to take you through how we get this equation and then how we reason our way through this. There's just a little bit of math. Stick with me. It involves properties of logarithms. If you're weak on that or don't remember, it basically doesn't matter. Just wait till the end, and then you can use the equation. So we derive this from the basic equilibrium of using some acid in the presence of water to get hydronium and A minus. Notice that this is not HA goes to H plus and A minus. Acids don't dissociate like this, especially maybe in the gas phase, but certainly not in water. In water, there is no such thing as H plus. It's all H3O plus. Water acts as a base to remove a proton from the acid. Okay? So I'm erasing this blasphemous writing here. So if we were to write an equilibrium constant for this reaction, it would be concentration of H3O plus times the concentration of conjugate base divided by the concentration of acid times the concentration of water. Now, probably in Gen Chem you were told that the activity of a pure substance is equal to 1, and therefore we can neglect water in this equilibrium expression. That is true, but we're using concentrations, not activities, and anything that I do now is going to be compensated for by those magic activity coefficients that you learned about and then forgot about. So just stick with me, because we're going to treat water just like a regular part of this equilibrium, except that at reasonable concentrations for acids and bases in aqueous solution, the concentration of water is constant. It's 55 molar, and it's basically impossible to change unless you start dumping in only a little bit of water and mostly some organic solvent. So for the most part, we're going to treat the concentration of water as constant, which means we can move it to the other side of the equation with the equilibrium constant to get this new, slightly more simplified expression like this. And we're going to define that new constant as Ka, which you were told is an acid dissociation constant, which is fine except for the acids don't dissociate in water. Water removes a proton from the acid. Okay, good so far. So now we're going to use a few definitions. We're going to define pKa as the minus log of Ka. We do that by analogy to the fact that delta G equals minus RT ln K, such that large equilibriums have negative pKa values. Large equilibrium constants, favorable Kas, have negative pKa values. We're also going to define pH as the minus log of H3O plus, such that Really low pHs mean high concentration of hydronium. Then we will take the minus log of both sides of our equilibrium expression, minus log of Ka, which equals pKa, and then minus log of H3O plus times concentration of A minus times, or divided by concentration of HA. Then we're going to use logarithm properties to separate out some of these things. And this is essentially the Henderson-Hasselbalch equation, or a form of it, but we're going to turn it into the useful form that we will use. We're going to subtract pH from pKa, and that's going to equal the log of the ratio between the acid concentration and the conjugate base concentration. So if you don't like the math and you don't like the derivation, you need to come back to us here and start paying attention again. This is the form that we will use. So our assumption is going to be that in aqueous buffered solution, the concentration of whatever acid we're interested in is going to be much lower than the concentration of buffer components, such that we treat the pH as constant, and the thing that's going to vary is this ratio. So let me give you an example where this is important in biology. So I'm drawing a very, very detailed structure of a protein, right, which is this great squiggly mark. And I'm going to draw some of the amino acid side chains. This one is called lysine, and it has a positively charged ammonium group. And the proton there, the acidic proton there, has a pKa of 11. You don't need to memorize that. I just happen to know it. There's another side chain that looks like this. This side chain is called arginine, and the acidic proton there has a pKa of 12. I'm writing the one-letter codes for those of you that know them. If you don't worry about it. I'm only writing the R because that allows me to tell my favorite amino acid joke, which is, what is a pirate's favorite amino acid? And it's arginine. Yeah. Nah. Charity chuckles are appreciated, but actually groans are okay too. So we have some amino acid side chains have carboxylic acid groups. That pKa is around 4, or anywhere in the 4 to 5 range. There are amino acids. I'm making stuff up here. There are amino acids that have aromatic rings, and then that have an OH group. That's a phenol. Its pKa is 10. And there's even a weird one that looks like this, and the pKa of that acidic proton is 6. Okay, I've drawn all of these in their acidic forms. Now we're going to compare the pKa's to physiological pH. Let's suppose we're in the cytoplasm, or in the extracellular matrix, or in the blood, and pH is 7.4. So, starting with the ammonium group, pKa of 11. I'm going to take 11 minus 7.4. That is, and here you don't even have to do subtraction. You just have to be able to do greater than, less than. 11 minus 7.4 is a positive number, right? Which means that this ratio has to be greater than 1, less than 1, or equal to 1. Greater than 1, right? If the logarithm is positive, the number inside has to be greater than 1. So that means the acidic form dominates. So at pH 7.4, what does this side chain look like? Well, it's positively charged, and it's in its ammonium form. All right? That's important because charged groups tend to be on the surfaces of proteins instead of in the interior, where on the surface they're exposed to water and can interact with water via hydrogen bonding or via counterions. A positively charged amino acid can interact favorably with a negatively charged amino acid, and that really does affect protein folding and structure. So if you're looking at a protein sequence and you see lysine, right away you should be thinking charged, positively charged at physiological pH. Similarly, with this arginine side chain, pKa 12 minus 7 is also a positive number, which means the positively charged form dominates at physiological pH, and that group will also be positively charged. How about the carboxylic acid? 4 minus 7 is negative. That's all you need to know. So the conjugate base form dominates, and it'll be negatively charged. 10 minus 7 is positive, so that one's going to be neutral. It's going to exist in its protonated form. How about 6? How about pK of 6? 6 minus 7 is negative, but not by a lot. A log of a ratio where that logarithm equals 1.4 probably tells you that, 6 minus 7 is negative 1.4. That tells you the conjugate base form dominates, but it may be a situation where you've got maybe 90% of the conjugate base form and maybe 10%. I'm just sort of spitballing here. Spitballs in the day of COVID, that's really disgusting. I think we should change that phrase. I got nothing, so we'll work on that. Probably 90% conjugate base form, around 10% acid form. This is actually really important for biology because this side chain is called histidine. It has a one-letter code of H. It is involved in tons of enzyme active sites where it needs to be both in its conjugate base form and it needs to have reasonable access to the conjugate acid form. You'll see some examples of that a little bit later. But that balance between is it in its acid versus base form is really important in biology and biochemistry. So, moral of the story is be prepared for me to draw an obscene and heinous molecule with lots of acidic protons on it and be able to pick out the acidic protons, look up the pKa on a pKa table, and then answer the question, if the pH is this, what is the total charge on that molecule? And to answer that right, you've got to get each of the acidic groups in its proper form at that pH. Let me just warn you of a mistake that people make with amino and ammonium groups. Amino groups are a bit challenging because they have two relevant pKa's. Going from the neutral amine to the negatively charged amide has a pKa of 38. And so some people, when I give these questions on a test, I will usually draw groups in their neutral form, whether that's the acid or the base form, and then you have to determine what the correct form is at physiological pH. Some people see this and they go to the pKa table and they look at this and they say, okay, pKa of 38, therefore we don't have any of the negatively charged groups at pH 7, which I agree with. That's totally true. Nevertheless, this neutral form is not the dominant form at pH 7 because there is another acid-base equilibrium to be aware of, which has a pKa of around 11, right? And that tells you that at pH 7, you also don't have any of the neutral amine. You have the positively charged ammonium form. People make that mistake all the time, and then they come to my office and say, well, I thought the pK was 38, and so here I am warning you. I'm having the same discussion I would have with you individually in my office about this problem if you were to get it wrong on a test. All right. Anything else we need to say about that? Yeah.
So I could be wrong, but I thought
Okay, so this is a question. How do I know whether I should remove one proton versus all of them? You do a acid by acid comparison based on the pKa versus the pH. Is that? The other way that your book may ask this question is, can this base remove this proton? And in that case, you set up the equilibrium between your base and your acid one by one, and you measure the pKa of that of the base of the conjugate acid of the base on this side, and the pKa here, and the strongest acid loses its proton.
And so would that mean that if all of the protons were equally acidic, that they would all be?
Yeah, I mean, we're assuming in questions like that, like you're adding an excess of base, and the question really is. The reason we think about it that way for synthetic chemists is that we're asking which protons could interfere in this reaction. Do you remember we talked about protecting groups for alcohols, and that the alcohol proton was too acidic to justify leaving it unprotected in the presence of a strong base? Well, what if there are multiple alcohols? Well, we'd still want to worry about all of them because we would need to protect all of them. So that's sort of the reasoning. Others? Yeah.
Yeah. So on the structure second from the right on the protein chain, it says it's neutral. Why is that? Because you have a pK of 10, right?
Yes, that's because its acidic form is neutral. The conjugate base form is negative. So at pH 7, 10 minus 7 is a positive number. So yeah, the acidic form dominates, which just happens to be neutral for that acid. Okay. Yeah. A couple other acidic groups that I want to mention that you may encounter in biology. I don't want to belabor the point too much. This is phosphoric acid. It has 3 equivalent OH groups, H3PO4. The first proton to be removed, and it doesn't matter which one because they're all equivalent, generates this anion, which only has one negative charge. We'll call that a monoanion, and that's H2PO4 minus. Once you already have a negative charge, the pKa of these two protons increases. It gets harder to remove, to go from having one negative charge to two, because the negative charges repel each other. And this is why the pKa of the first proton to come off is around 2.2, but the next proton to come off has a pKa of 7.2. And the conjugate base of the monoanion is the dianion, and that's HPO4 minus. And as you can imagine, removing the third proton is even harder. That pKa is 12.3, and that would get you to PO4 minus. Okay, so phosphate is an important molecule in biology. So let's see what form the phosphoric acid should take at pH 7.4. So we're going to do this pKa by pKa. Should this proton, the first proton, still be there at pH 7? pKa minus pH, negative number. So conjugate base form dominates. Okay, now let's do this one, the monoanion. pKa is 7. 7.2 minus 7.4 is a negative number, but a small one. Basically 0. That tells you when pH minus pKa is 0, for what number does a logarithm equal 0? 1, right? So if pH equals pKa, you actually have about a 1-to-1 mixture of acid and conjugate base forms. So at pH 7, you probably have a 1-to-1 mixture of the monoanion and the dianion. How about the last proton, pKa 12? Is that on or off at pH 7? On. 12 minus 7 is a positive number, which means the acid form dominates. So the biologically relevant forms of phosphoric acid are these two. Your biochemistry and biotexts likely ignore the charges in the acid-base equilibrium, and they call this PI, or inorganic phosphate, as though inorganic were a pejorative, like an insult. Oh, that phosphate's inorganic. I mean, the difference is it's not attached to an organic molecule. Where does this show up in biology? Let me just give you a couple examples. When we start to talk about glycolysis, we will encounter a molecule called glucose 6-phosphate. And glucose 6, oh, that is a terrible chair. Let's try that again. Slightly less terrible. There we go, that's better. Glucose 6-phosphate, we can draw in this form, but that's not its physiologically relevant form. This functional group is called a phosphoester because we've replaced one of the OH groups of phosphoric acid with an alcohol group, an OR group. So by analogy to carboxylic acids and esters, this is a phosphoester. The first pKa is around 2, and the second pKa is around 7. So at physiological pH, what should be the dominant form? Well, it should probably be a 1-to-1 mixture of monoanion and dianion. And so sometimes I will abbreviate the phosphoester as OPO3 2-. Another place you will encounter this is in DNA. Have you ever wondered why DNA and RNA have acid in their name? Deoxyribonucleic acid, or just ribonucleic acid. I'm focusing in on the sugar phosphate backbone of DNA. This is a phosphodiester, and I'm going to show it in its neutral acidic form. This is why, actually, DNA is called an acid. You may have been perplexed to call it DNA, it's an acid, but then we spent all this time talking about bases and base pairs. Well, the bases do have basic groups on them, but the acid comes from the backbone. And the pKa of that first OH group is still around 2. So at what's going on here? Why can't we, the little thing on the tip of the Apple Pencil was coming undone, so we fixed it. You know, one time, I tend to fidget, and one time I was in the middle of a lecture playing with this, and I dropped it and it bounced, and it actually fell in the tech podium. You can come see it after class. There's a little bit window there that the thing fell in, and it was a disaster. Somebody from the back of the class ran up and let me use the tip of their Apple Pencil, and that kid got an A, of course. I'm just kidding. Really just kidding. There's no purchasing of grades or anything like that, but I was really embarrassed. So I'm going to learn my lesson and stop fidgeting. Anyway, at physiological pH, that proton should be gone. And so the sugar phosphate backbone of DNA is negatively charged. That's really important for DNA structure. There are counter ions and water molecules that line the sugar phosphate backbone, and transcription factors that need to bind DNA to prepare it for transcription have to deal with that negatively charged sugar phosphate backbone. Okay. So I think that's all we want to do on acids and bases. Any other things you want to ask? Okay. So now we're going to move on to chapter 16, and this is chemistry of carboxylic acid derivatives. We're going to start with the hard stuff, which is with the carboxylic acids themselves. And then we'll go to some of the easier stuff. We'll spend most of our time on the hard stuff, little time on the easy stuff that you can get on your own. Use the study guides to guide yourself in terms of what you should be studying on your own. And we'll try to deal with the most important things in class. So suppose we want to turn a carboxylic acid into an ester. We've kind of got a problem because the nucleophile, the alcohol, is neutral. And we've said the neutral alcohol is likely not strong enough to attack the carbonyl carbon to give you a negatively charged oxygen directly. Okay, well, what if we instead used the negatively charged alkoxide, the deprotonated alcohol? The problem with that is that carboxylic acids have an acidic proton, pKa of around 5. So instead of attacking the carbonyl carbon, it's actually the negatively charged alkoxide is simply going to remove a proton from the carboxylic acid because the conjugate base of the carboxylic acid is much more stable than the alkoxide, pKa 16 versus 5. It's easy to see that you just deprotonate the carboxylic acid and sort of be dead in the water. So when we're in this situation, as we were before when we're in a situation where we have a neutral nucleophile that can't attack directly, we've used acid catalysis. So we're going to use a catalytic acid, and that's going to first protonate the carbonyl oxygen, which activates the carbonyl group as an electrophile. And the reason that works is because once you put a proton on the carbonyl oxygen, you make this resonance structure where the positive charge is on the carbon even more reasonable. So the protonated carboxylic acid is more electrophilic, and now the neutral alcohol can attack. This should look familiar to you in the sense that these were also the first steps of acetal formation, only the carbonyl compound was an aldehyde or a ketone. But same basic steps, we protonate the oxygen and then an alcohol attacks. The alcohol attacks the pi star of the carbon-oxygen pi bond, the antibonding orbital, at the carbon, because that's where the electron poor spot of this molecule is. We break the carbon-oxygen pi bond to get a tetrahedral intermediate. And this should look familiar too. The only difference is that this group is not a proton or another R group. So now that we have a tetrahedral intermediate, our job is to identify whether there are any good leaving groups. And in this case, there are. There is a good leaving group. It's the protonated alcohol. And that's a problem because ultimately we want that alcohol group to stick around. So we need to do the proton transfer shuffle to change who's the good leaving group. Fortunately, we have the conjugate base of our acid to assist in this process, so we'll remove a proton here. That takes us to an intermediate where no one is a good leaving group. And then we can protonate one of these, I don't care which one of these oxygens using our catalytic acid, thereby changing one of those OH groups into a positively charged ROH2 group, which can leave as water. So now, through that series of steps, we have changed who the good leaving group is. Now that we've got a good leaving group, electrons will kick down and leaving group leaves. That's reasonable for a neutral oxygen to kick off a positively charged leaving group, because what happens when we kick off that leaving group is that we get another positively charged intermediate. And these two are approximately similar stability, so it's reasonable. Notice that water was the leaving group there. That's going to be important. Sorry. And then all that's left to get us to the ester is for the conjugate base of our acid to come in and remove the proton, and that regenerates our catalytic acid and then also gives us the ester. Okay? So that should look really familiar to you. The steps are exactly the same as acetal formation until we get here. When we get to this point, we don't have another alcohol come in and attack. That kind of thing, such molecules are known in organic chemistry, but we're not going to worry about them. So the idea is protonate the carbonyl oxygen, neutral nucleophile attacks, proton transfer shuffle to change who's the good leaving group, electrons kick down, water leaves, and then we deprotonate the oxonium ion to get to our ester product and regenerate our acid. As with acetal formation, this is an equilibrium, so we're going to drive this by removing water. Okay? Now this process has a name. This is called Fischer esterification. It's one of those reactions that's named after the dead guy and also what the reaction does. Dead guy, process, and ification is how we basically name organic reactions. Now, some of you are in 353. Yes, sort of. Have you done the esterification reaction? Okay. How did you remove water? Use a salt. It's either sodium sulfate or mag sulfate, maybe calcium chloride. And then silica. Silica absorbs water, so does that salt. So you're removing water from the equilibrium. That drives the reaction forward. The ester you made, was it banana smelling? Yeah, so a lot of esters, low molecular weight esters, actually are very fragrant and are used in flavoring and other places where smells are needed. Now, it turns out that we can do the reverse of this reaction. It's called ester hydrolysis. And ester hydrolysis under acidic conditions involves all of these same intermediates, only we're going to use an excess of water to take us back to the starting materials. So this is not a separate reaction to hydrolyze esters under acidic conditions, just as hydrolyzing acetals under acidic conditions was not a separate reaction. You're going to use an excess of water to generate the acid and the alcohol. I'm not going to walk you through that mechanism because you have all the intermediates here. You just need to be able to push the arrows backwards. Okay? So give that a try. If you're struggling with it, talk to one of your TAs. They can help you with it, or come talk to me. When we meet next time, we will talk about how our ancestors made soap using chemistry that we'll learn about. Yes, sir.